everyone, my name is Pei-Chen Peng. I'm a postdoc at Cedars-Sinai Medical Center. First of all, I would like to thank for this great opportunity to share our work with you. Today, I'm going to talk about how we integrated GWAS risk SNPs with functional genomics to identify causal SNPs of ovarian cancer. Genome-wide association studies, or GWAS, have uncovered many significantly associated SNPs. The next challenge in this field is to find SNPs that cause the disease. And this challenge has been complicated by two problems. First, the linkage disequilibrium, or LD. Let's say there are two SNPs in perfect LD. They will have the same association statistics, and it's therefore unclear whether one is more important than the other. And second, a SNP with the greatest p-value may not necessarily be the causal SNPs as shown here. During the past decade, the ENCODE project and other databases have collected genomic and epigenomic data. These enable us to annotate the SNPs and characterize regulatory landscapes of the risk regions. In this presentation today, I will mainly discuss two parts. First, how do we identify significant functional annotations for ovarian cancer? And second, how do we apply a probabilistic model to prioritize causal SNPs? The disease we studied here is ovarian cancer, which is the most lethal gynecologic cancer in the world. And this ovarian cancer GWAS dataset has around 26,000 cases and 105,000 controls. The largest subtype in ovarian cancer is HGSOC. And from the GWAS, we identified 44 risk regions. The first question we asked was where the risk SNPs reside in. Our first attempt was to annotate SNPs with the 24 functional regions identified from ENCODE, such as 3' UTR, 5' UTR, and coding regions, and so on. And we performed SNP enrichment analysis. And instead of counting the presence of SNPs intersecting in each region, we estimated the partition SNP-heritability. And the SNP-heritability shows how much variation of the disease in the population can be accounted by SNPs variation. So this way we can take the linkage disequilibrium into account. Okay, so here's the result. Significant enrichment were colored in orange, and non-significant enrichment were in blue. As you can see from figure here, we observe a strong enrichment from the 3' UTR regions in both all non-mucinous and HGSOC subtypes. We also observe significant signal in regulatory elements like enhancers and promoters. And since ENCODE data was from common cell types, we next asked if the enrichment will be stronger if the ovarian cancer-related cell types are being used. To answer that, we generated a catalog of ovarian cancer epigenomic annotations. 18 ovarian cancer-related cell lines were profiled by RNA-seq and ChIP-seq for 4 histone marks. We trained a chromatin model to map the histone marks into 8 chromatin states as listed here. Then we repeated the partition heritability enrichment analysis on these ovarian cancer related chromatin states. Our results shows that the most significant enrichment were from the active promoter region, color coded in the dark purple here. And as a comparison, we also observe enrichment in the ENCODE promoter regions for the general cell lines, but the extent of enrichment is much higher in ovarian cancer related cell lines compared to the ENCODE common cell lines. The second part of this study is to use a probabilistic model to integrate significant functional annotations and to prioritize credible causal SNPs. We applied the Bayesian-based model called SPAN-TOR to identify likely causal SNPs in each of the 44 GWAS risk regions. Here we show the results for HGSOC SNPs prioritized by the PAN-TOR model. We found several of their nearby genes have been reported in literatures. So for example, the PAX8, it's a well-known transcription factor overexpressed in ovarian cancer. PVT1 has shown to promote the ovarian cancer progression. On the right-hand side, the SMC2 and BABM1, they have been linked to the EQTL in GTEx ovary and TCGA ovarian cancer databases. To conclude, here in this study, we demonstrated that integrating GWAS and functional genomics can help identify new candidate causal SNPs. And we also emphasize that the importance of using disease-related annotations in SNP prioritization. With that, I would like to thank my mentor, Simon Gaither, and the lab. And this is a collaborative work across many institutes. We appreciate everyone who put effort in this project, either on data acquisition or computational analysis. Last but not the least, I would also like to thank the patients and their family for their support in this project. Thank you.